Hello students, I am Dr. Maria Siniola Fernandez, Assistant Professor in Botany, St. Joseph Vaz College, Kortalim, Goa. Welcome to the program Bachelor of Science, Subject Botany, Semester 1, Course Title BOC 101, Course Code BOC 101, Course Title Biodiversity 1, which includes microbes, algae, fungi and bryophytes. Title of the unit is Bryophyta. Module name Reproduction in Funaria. We will study the vegetative method. In this module, we will study the different methods of vegetative reproduction in Funaria. At the end of this module, you will be able to describe various methods involved in the vegetative reproduction in Funaria. So, we will study the different methods of vegetative reproduction in Funaria. It is the gametophytic stage of Funaria that reproduces vegetatively by various methods. The following methods, the first method is the multiplication of the protonymal stage. In this method, the primary protonema, it breaks into short fragments of living green cells by death of cells at various intervals. Now, at each detached fragment of the cell, it consists of green cells or several cells that grows into a new protonema, which bears a fresh crop of leafy gametophyte. This new protonema, which has formed from this protonymal stage, it has the capacity to grow into a new fresh gametophyte. Second method is secondary protonema. The filamentous protonema is formed from any cell of detached injured portion of stem, leaf and rhizoids. It is known as the secondary protonema. The third method is by bulbils. Bulbils are the small resting buds which are developed on the rhizoids. When there is suitable condition or when the onset of this suitable condition for the growth of this bulbil, it produces protonema of moss gametophores as lateral buds. Here in the diagram you can see the gametophyte. This is the gametophyte which shows the secondary protonema and bulbils. We will see what is it. From this gametophyte, this filamentous protonema is formed. It can be formed from any cell of detached injured portion of stem, from the rhizoids, or it could be even the leaf. So, you get this secondary protonema, and this secondary protonema has the capacity to grow into a new gametophyte. Other method what we have seen of the vegetative reproduction is by bulbils. Now, these bulbils, they are small rounded structures which develop on the rhizoids. So, they are underground, you can see that, and when there is suitable condition for their growth, this bulbil, they will produce or grow into a new gametophyte as lateral buds. The fourth method of vegetative reproduction of Funaria is by gemmae. Now, gemmae are the terminal cells of the protonymal branches which forms this small thin walled bud like chloroplast which bears these structures called gemmae. So, once this gemmae is in the functional state, they get detached from the parent plant and they produce this new protonema, and this protonema has the capacity to grow into a new gametophyte. The fifth vegetative method is by apospory. Apospory is from the formation of from the green protonymal filaments which arise from the unspecialized cells of various parts of sporogonium. The cells, the green cells, they develop from the unspecialized cells of various parts of sporogonium. They are green in color and they have the chloroplast. So, these bear lateral buds. This green protonymal filaments, they bear lateral buds which develops into a leafy gametophore. Now this leafy gametophores or gametophytes which are produced directly from the vegetative cells of the sporogonium without the intervention of the spores is called apospory. So the various methods which are involved in the vegetative reproduction of Funaria, first is by the multiplication of the protonemal stage, where the cells, the green cells, they multiply of the protonemal stage and they have the capacity to grow into a new gametophore. Second method what we have seen is by the secondary protonema, the filamentous protonema which arise from the leaf, stem, or they can be even from the rhizoids. So they have the capacity to grow into a new gametophyte. Third method what we have seen is by the bulbils. Now bulbils, I told you, they are the resting buds, okay, which are found on the rhizoids, and when there is suitable condition they will produce into a new gametophyte. The fourth method is by gemmae. Okay, gemmae are nothing but the terminal cells which are produced on the protonemal branches, which are of small thin walled structures which bears chloroplast, and they get detached and they can germinate into a new protonema. The fifth method what we have seen is by apospory. Apospory takes place from the unspecialized cells of various parts of sporogonium, and this produces the lateral buds which develops into leafy gametophore, and the leafy gametophores which are produced directly from the vegetative cells of the sporogonium and where there is no intervention of the spores is known as apospory. These are the references. Thank you.